A little announcement. Well, first of all, welcome to day 4. You've survived the first 3 days and will continue to survive as we go along. How many of you would like to work on Friday night till about 10 o'clock in exchange for getting out a little early on Saturday?
Sure.
Actually, we're not going to work Friday night. We're going to give you Friday night off. That was a joke. And because we're giving you Friday night off, we have a little something planned. So I will let Kalani tell you about the potential plans. Well, that's a plan.
So historically, we usually all go out, or we don't all have to, but many of us go out together on Friday. So I'm here just to get a show of hands of how many people think they'd like to come out with us on Friday so that I can start looking into some reservations. Usually what we do is, so what happens on that day is you guys start running a gel right before dinner, and then you finish up at 6 o'clock, and then we finish running your gel and we photo it for you, and then we usually meet you somewhere at 7 p.m. So what we usually do, the staff, is we go somewhere and get dinner at 7 p.m. You are welcome to wait and have dinner with us at 7 p.m. But if you think you're gonna be hungry, you can get dinner before, whatever. So we want a show of hands of people who think they would like to come with us for dinner at 7. But then if you want to come out with us later, we usually also go to another bar after that. So if you think you're gonna miss out if you don't come or whatever, there's also a second location, and we'll share phone numbers and stuff to make sure you catch up with us. So, who thinks they would like to come out for dinner at 7 p.m. on Friday? Show of hands. Ooh, lots of hands. Okay, I'm gonna count. 1, 2, and if you want to double count me too, to make sure I do it right. 1, 2.
I got 29.
Oh, put them back up.
That's fine. That's close enough for government.
Well, no, but we also, there's us, and we have questions within us, so we might.
No, but it's over that 25 number, so.
Oh, shoot, you're right.
Yeah, so I
Okay.
think it doesn't matter. So I think that's,
I have,
fine.
well.
Da-da-da-da-da.
Okay, that's more than a certain cutoff number, so I have to go back to the drawing board, and I will probably have to take another poll at some point just to reconfirm stuff. Also, just so you know, I meant to say this, but I forgot. We don't pay for anything, so you are on your own payment-wise. We only pay for ourselves. This is not included in the course budget. Okay.
Allison would have a heart attack.
Yeah, I will make some phone calls, and then I will probably, unfortunately, do a secondary poll later. But I'm glad that there's so much interest. We'll love to hang out with you all. Okay, that's it.
Yeah, it's always fun. We go to a restaurant downtown that's got a wide variety of food, so everybody's.
Well, although that, we'll see. Things might be shifting, because there's a lot of you this year. So, all right, I'll be back at, not now, but maybe another lecture to do a similar poll. Thank you, guys. Thank you.
So, an important reason for getting a count is not just for the restaurant, but it's also so I can tell dining services not to make very much food on Friday night, because it won't be there. So that's good to know. Yes? Oh, no. Sorry to disappoint you. No, Friday night you get off, not tonight. Anyway, so Friday night you get off, pretty cool. And then Saturday, as I said, we end by 3 o'clock, and then you get 41 hours of freedom until we're back there Monday morning. So so looking kind of cushy coming ahead. The next couple of days are not particularly easy, but then Friday, short day, Saturday, short day, Sunday, day off. So there's a light at the end of the tunnel for all of you. Did you get that? Yeah, okay. All right, a lot going on today. We're afraid you're getting a little bored maybe, so we're adding in another experiment tonight. So experiment 6 starts tonight. This is our human genetics experiment. experiment where we're gonna have you isolate DNA from your cheek cells, and we're gonna do DNA fingerprinting, and we're gonna do this phenotype-genotype experiment with tasting. So that's a lot of fun. Mostly today, we're working on experiment 5, and experiment 3, and a little bit on experiment 1. This is a pretty light day for experiment 1. At any rate, this morning, we're working on 3, 1, and 5. So I'll take you through the protocols. It's not complicated, actually. So even though we've got 3 experiments in the same period, it's not bad, I promise. Okay. All that's going to come later. I'm going to start this morning by talking to you about gene expression in bacteria and in eukaryotes in more detail, because we need a little more detail going forward for making this gene fusion that we're talking about and getting our gene fusion expressed in E. coli. So I need to give you some more detail. So we're going to look at bacteria first, and in bacteria we work with E. coli. So specifically I'm talking about E. coli here, but it works similarly in all bacteria. So even though I'm talking about E. coli, this is the general situation that we see in bacteria overall. So my double railroad track here is our DNA in purple. As usual, the RNA is shown in red or pink here. So I want you to focus on this part of the diagram here from here up. Ignore the DNA sequence down here for now. In bacteria, remember, we transcribe the gene and then we directly translate it. There is no pre-mRNA, there is no RNA processing like we see in eukaryotes. So remember, no processing. That means no 5' cap, no intron exon splicing, no poly A tail. Okay? In bacteria, the gene just gets transcribed and then it's immediately translated. In fact, because in bacteria there's no nucleus, transcription and translation are happening in the same compartment. A bacterial cell is really just a bag, and just a single bag. There are no bags inside the bag, right? So in eukaryotes, the cell's a bag, but there are bags inside the bag, right? There's a bag that's the nucleus, and there's bags that are the mitochondria, and if you're a plant, there are bags that are the chloroplast, right? In bacteria, none of that. So it's all happening in one place in the bacterial cell. And because of that, we say that in bacteria, right here we say transcription and translation are coupled. So transcription and translation are happening in the same compartment. And so what actually happens is that the gene starts getting transcribed into the RNA. And once the gene starts getting transcribed into RNA, ribosomes can load onto the RNA and start translating immediately. So ribosomes get on the messenger RNA and start translating before transcription is even completed. Okay? So they're coupled. And that means that things have to be simpler in bacteria. In eukaryotes, transcription happens in the nucleus. That's where the chromosomes are, right? So transcription happens in the nucleus. The processing happens in the nucleus, and I'll go through this again when we do eukaryotes. The processing, that is the addition of the 5' cap, the intron exon splicing, and the addition of the poly A tail, that all happens in the nucleus. And only when the transcript is completely processed and is a final mRNA does it then get shipped out to the cytoplasm for translation. So transcription happens in the nucleus, translation happens out in the cytoplasm, And because of that, in eukaryotes, we say that transcription and translation are uncoupled, because they're happening in separate compartments. So you don't get any of this business in eukaryotes where translation starts before transcription is even finished. That only happens in bacteria. Yes, sir.
What about the transcription and translation in the mitochondrial DNA?
In mitochondria and in chloroplasts, it happens just like in bacteria. So far, so good? Yes, sir.
Are they specialized systems that could be different, like could be different cells, like RNA cells for those specialized cells?
There are specialized cells that sometimes do things a little bit differently. So this is generalized for bacteria, what I'm telling you about now. But in archaea, for example, things are a little bit of a hybrid. So archaea don't have a nucleus. So in archaea, transcription and translation are coupled because there's no nucleus. But then there's some details of transcription and translation that are more like eukaryotes. So it really depends on what kind of cell you're looking at. It also depends on if you're looking at, you know, the nucleus in eukaryotes or the mitochondria or the chloroplast. Things are happening slightly differently in all those compartments. Okay, so what I'm talking about here is gene expression in bacteria, but it works very similarly inside eukaryotic mitochondria and chloroplast. Okay. So far, so good? Okay. So let's take a closer look at how this happens. Remember, I showed you before that transcription has to initiate at a promoter, and that a promoter is the binding site for RNA polymerase, right? And is the signal for where transcription is supposed to start. So I want you to imagine the E. coli chromosome is 4.6 million base pairs. It's a big circle. It's got a few thousand genes on that chromosome. And those genes are scattered around that circular chromosome. And the cell has to know where to start transcribing and where to stop transcribing. It can't just do it, you know, willy-nilly, right? And so the signal for where to start transcription, which would indicate the beginning of the gene, is the promoter. And so the promoters are critical so that RNA polymerases know where to begin transcription. Now, a promoter is somewhat more complicated than you might have imagined from my first description. So I'm going to show you in a little more detail here. The promoter has two critical regions in it, and those two critical regions are called the -10 box and the -35 box. And I've actually shown the sequence of a -10 box and a -35 box down here. This is an example of a -10 and -35. They're not all the same. So this is from a particular gene in E. coli. If you look at a bunch of genes, you see some variation in the -10 box, some variation in the sequence of the -35. So they're not all the same. The distance between the -10 and -35 box is also important. It's almost always between 16 and 22 base pairs. So you've got the -10 box, you've got the -35 box, and then a region in between. And what I want you to imagine is what the RNA polymerase does is it locks onto the promoter at those two sequences. So the polymerase, boom, grabs onto those sequences, and that's how it recognizes the promoters. These proteins that bind to specific DNA sequences, the same is true for the restriction enzymes we were talking about yesterday. These proteins that bind specific DNA sequences actually have fingers that reach into the DNA helix and contact the bases and can actually, in a sense, read the sequence, right? They can reach in and go, oh, this isn't the one I want, and then it lets go and goes somewhere else, right? If it reaches in and finds the right sequence with its little fingers, then it sticks, and that's what's going on with the RNA polymerase here. It's binding, it's a big molecule. The RNA polymerase is big, it covers this whole promoter region, but it's got two regions of fingers that reach in and, in a sense, read the sequence. So those of you who've studied this at all might know about zinc fingers, and so it's these zinc fingers that are reaching and contacting the bases. So that's pretty cool to think about. The other thing is these things are happening incredibly fast. So, you know, the polymerase binds to a sequence, reaches in, oh, that's not right. Binds to another region, reaches in, that's not right. Binds to another region, reaches in, oh, that's right. That sounds incredibly slow and inefficient, and it's kind of random, but it's happening so fast that the system works, okay? So, that's what's going on at the promoter. Now, once the RNA polymerase binds to that sequence, the next thing it does is it pulls those two strands apart. So the RNA polymerase pulls the two strands apart, and once it pulls those two strands apart here at the promoter, now it can start doing transcription. It can start transcribing the bottom strand here, the template strand. And so what the RNA polymerase does then is it moves along the DNA, separating the DNA as it goes, and copying that template strand into the messenger RNA. So imagine the RNA polymerase binding here, and then it starts moving down, and as it's moving down, it's separating the DNA. Behind it, the DNA goes back together, okay? So it's only separated where the RNA polymerase is, and it's moving down the molecule and doing transcription. And once it gets to the terminator sequence, then the RNA polymerase, that's the signal for it to come off, and it releases. And when it releases the mRNA, and now that mRNA is free in the solution. Okay. Now, where exactly does the RNA polymerase start transcribing? It transcribes about 10 nucleotides down from this -10 box. It transcribes starting at +1. And so here I've shown the -35 box, the -10 box, and then +1. +1 is almost always an AT base pair, and that's where transcription starts. So the first nucleotide in most transcripts is an A, because we start transcription at this AT base pair. So here I've shown the actual sequence of the -10 and -35 box. The way these things get their names is that the middle of the -10 box is about 10 nucleotides upstream from +1, and the -35 box is about 35 nucleotides upstream from +1. Okay? So again, looking at this diagram down here, imagine we've got this big beach ball of an RNA polymerase, and it binds here, grabs onto those two sequences, separates the two DNA strands, and then starts transcribing at +1. The RNA polymerase is big enough that it's covering this whole region, the -35 box, the -10, the +1. It's sitting on this whole region here, and it can separate this entire region that I'm indicating here. Okay? And then it starts moving down and transcribing the entire region. And it won't let go until it gets to that terminator sequence. The spacing between the -10 and -35 is important. The spacing between the -10 and the +1 is important. It's usually 5 to 7 base pairs. And then after +1, somewhere down here, there's another important feature, and I've shown it here. This is blown up, you know, we're blowing up this region here as this big region here, right? The next feature down here that's important is the ribosome binding site. So that's right here. The ribosome binding site is encoded in the DNA downstream from +1. So here's +1, and here's our ribosome binding site. It's about 25 base pairs downstream from +1. When this gets transcribed into RNA, the mRNA, starting at +1, the ribosome binding site has this sequence, AGG, AGG, and that sequence binds the ribosome. That's how the ribosome knows where to bind. And then what it's going to look for is a start codon 6 to 10 base pairs downstream from that ribosome binding site. So the ribosome is gonna bind to this region, and it needs to find that AUG within 6 to 10 base pairs. If there's no AUG there, then it's not gonna translate. But in a gene that's intact and hasn't been mutated, downstream from the ribosome binding site will be an AUG. And that's how the ribosome knows where to start. Again, this AUG is going to be 6 to 10 nucleotides downstream from the ribosome binding site. If it's too far away, it's not going to work. Okay? Yeah.
I was just wondering, what's actually the force that lets like polymerases, ribosomes bind to it?
Sorry, say that again.
What's kind of the force that lets it bind to it? Because like it looks for the sides, but how? Like is it complementary? No, because then
So
you
the
would-
ribosomes are about half RNA and half protein. So the ribosome has within it, in bacteria, the ribosome has within it 3 RNA molecules called ribosomal RNA molecules. We're going to talk about this later today when we run your, when we look at your RNA gels. But there are 3 RNA molecules within the ribosome, and one of those 3 ribosomal RNAs actually hybridizes to this region, and that's how the ribosome recognizes the ribosome binding site. There's actually RNA in the ribosome that's going to hybridize to the ribosome binding site by base pairing, and that's how it knows where to start. Pretty cool.
Sort of a related question. So once it's bound, how does it know to actually what's driving it along the direction of the DNA and the transcript?
So the directionality. Let's talk about the promoter and the RNA polymerase first. The RNA polymerase is not symmetrical. So when the RNA polymerase binds to the -10 and -35, it's binding in a certain orientation, and it can only move that way. Okay. If you took this -10 and -35 and flipped it over, now the RNA polymerase would bind this way and it would go that direction.
Oh, okay.
Okay?
Yeah.
So it's really the orientation of -10 and -35 determines how the RNA polymerase binds, and that determines the direction it's going to go. So once it binds here, it can only go in that direction. The same thing is true for the ribosome. The ribosome binds here in an asymmetrical fashion and can only move that way. Yeah.
What do you mean by asymmetrical fashion?
So, how can I explain this? So if we look at the ribosome, the right side of it is not the same as the left side of it. So it's not a symmetrical molecule. And when it binds to the site here, it can only bind with its right side on this direction and its left side over here, and that means it can only go in that direction. Yeah. Okay? Yeah.
When you said the ribosome like hybridizes at RBS, does that even have a nucleic acid like actually hydrogen bonding?
Yeah, the 16S ribosomal RNA, I'll get into this later, actually hybridizes to that region. It doesn't need a helicase activity because the RNA is single-stranded. Okay? Yeah, it's not binding to the DNA, it's binding to the RNA. Yeah, no worries. Pretty exciting, huh? The kind of stuff that gets me going. I like this stuff. Any questions about that? All right, so.
Sorry, one more question.
Yeah, no
The
problem.
the actual sequence of the ribosome binding site, the -35, -10, can change from gene to gene, or
A little bit, right. So what you see is in another promoter, the sequence here might be TATGAT, right? So they're usually pretty close. This sequence is a really good sequence for binding RNA polymerase, but not all promoters are created equal. Some promoters are really good at binding RNA polymerase, and some are pretty lousy at binding RNA polymerase. But that's okay, that's what you want, actually. If you have a gene where you only need a small amount of that product in the cell, what you wanna have is what we refer to as a weak promoter. That is a promoter that's not gonna bind RNA polymerase very often, because you don't need a lot of transcript, because you don't need a lot of protein. On the other hand, there are some genes that have to be transcribed heavily and constantly in order to make enough product, and those genes will have what we refer to as a strong promoter that binds the RNA polymerase really efficiently. So for some genes you want a weak promoter because you don't need a lot of product, and for other genes you want a strong promoter because you need a lot of the product. Does that make sense? The closer you are to this ideal sequence, the more transcription you get. The further away from this ideal sequence, the less transcription you're going to get. But that's okay. Over the course of evolution, promoters have evolved to give the appropriate level of expression. Okay? Yeah.
So how many RNA polymerase in E. coli?
Oh, how many RNA polymerases in E. coli? So that do transcription, there's one, okay? Eukaryotes, which we'll get to in a minute, there are multiple RNA polymerases, but for transcription in bacteria, there's typically just one. That one, of course, there's a lot of copies of it. There's not just one polymerase floating around in the cell that has to do everything. There are thousands of them. And they're all searching the DNA for a promoter to work from. Yeah.
So with the RBS, it's like the bottleneck, the ribosome, like hybridizes, then
Yeah.
the top half.
Yeah.
starts moving to the
Yeah,
side.
so the ribosome has a large subunit and a small subunit, and what happens is the small subunit comes on first, and then the big subunit comes on. Yeah.
So,
How does the rate of translation or transcription compare to the rate of translation? Are they?
The rate of transcription is just a little bit faster than the rate of translation, so you don't have a problem with the ribosomes catching up and knocking into the RNA polymerase. So if you imagine the RNA polymerase traversing down the gene and making the RNA, right? And then the ribosome gets on here. If the ribosome caught up, it would knock into the polymerase, but that doesn't happen because the ribosome is moving a little slower.
So, the polymerases are gonna transcribe ribosomal, transfer RNA, protein coding genes. Okay, all of them.
All of them, right? So there's one RNA polymerase that does it all, right, in bacteria. A little simpler. I'm gonna draw you a cartoon over here to show you a little more accurately what this looks like in a physical sense. So here's our DNA, and here's the region where RNA polymerase has separated the two strands. Okay, so the RNA polymerase, without me saying anything else, you know where the RNA polymerase is. The RNA polymerase is right here. The RNA polymerase is sitting on the DNA here, and it separated the DNA in that region. Now the promoter, let's say the promoter is down here. Okay, so transcription started here at +1. The promoter's down there, transcription started down there. In this region is where the RNA synthesis is happening. And so the RNA is here, and it looks like this. Because transcription started here, that's the 5' end of our message. As transcription moves, the DNA double helix goes back together and kicks the RNA out, right? So as this thing moves along, this tail of RNA that's sticking out gets longer and longer, right? The RNA is only hybridized to the DNA at the region where the RNA polymerase is sitting. So this is our 5' end of our mRNA, here's the 3' end, and nucleotides are being added here. And as the polymerase moves down, this RNA is gonna get picked out, and we're gonna keep synthesizing by adding to the 3' end. Notice that our RNA is hybridized to the DNA on the template strand here. So I put my 5' and 3' on here. 5', 3'. 5'. So it's moving down in that direction. And notice that even though the region of hybridization here is small, it still has to obey the anti-parallel rule. The RNA is 5' to 3' this way, the DNA is 5 to 3 that way, it's anti-parallel.
It's just the same for prokaryotes and eukaryotes.
Same in prokaryotes and eukaryotes, right? So when I do eukaryotes, I'll show you something almost exactly the same. Yeah.
So what's actually driving the RNA polymerase down that chain? Is it some kind of chemical gradient that's following along that direction, or is it like it's like it's moving on a railway track. What's causing it to move?
Wow. I think we may need someone who's more of a biochemist than I am. It's an ATP consuming process. It's catalyzing the synthesis of RNA. Why does any enzyme keep doing what it's doing? It's an RNA polymerase. That's what it's supposed to do. It's a catalytic reaction. It's an enzyme. It's like any other enzymatic reaction.
Okay.
It's just momentum, man.
And if you want to know more, find yourself a real biochemist. Right? I'm a molecular biologist. Molecular biologists masquerade as biochemists. We're not really biochemists. And in fact, if you ask a molecular biologist if they're a biochemist, they'll go, oh God. Because we don't have that sort of training typically. Some molecular biologists are biochemists, I'm just not one of them. Anyway, so there you go, that's the best I can do. Any other questions about how this works? So imagine the RNA polymerase moving down, and this tail of RNA is getting longer, and because we're in bacteria and it's all in one bag, ribosomes can start loading on at the ribosome binding site here, right? Ribosomes can start loading on and start translating along the mRNA, and ribosomes translate along the mRNA 5' to 3'. So a ribosome can get on here and start translating. And as this thing moves further down, and this ribosome moves further down, another ribosome can get on and start translating. So as this thing moves down the gene, you might have 50 ribosomes on here doing translation, just getting on one after another onto the ribosome binding site and starting translation. And if you look in the electron microscope, if you look in the electron microscope, you can actually see a line that represents the DNA double helix, and then you can see these thinner lines coming off that are the RNA. So the thinner lines coming off that are RNA, and then you can see these little blobs on the RNA, which are the ribosomes. So you can actually see this in the electron microscope. If I remember, I'll find one of those images and show you. It's pretty cool because you're kind of seeing it in action, which is nice. You only see this. So this would be the DNA. This little thin line would be the messenger RNA, and these blobs are the ribosomes, right? You only see this in bacteria. You don't see this in eukaryotes, right? Because remember, in eukaryotes, this process of transcription and the process of translation are uncoupled. In some images, you can actually see the protein starting to come off the ribosomes, right? and so, you can actually see a fair amount of detail in some, electron micrographs. Whew. Okay. Let me, go back here and remind myself if there's anything else I want to tell you about. We did a pretty good job here. Now you know everything you need to know, for our purposes for transcription in eukaryotes and translation. Two more things that I want you to think about. One is that translation is going to start at this start codon, and the first amino acid will be methionine. We haven't looked at the genetic code yet, but we will. So that's one thing we still have yet to do. And the other thing I need to do is to contrast this to eukaryotes where things are much more complicated. Now, I don't want to fool you into thinking that this is all the detail. So in bacteria, there's often some other things happening here at the promoter. So, for example, in the lac operon, which you may or may not have heard of, but this is a famous set of genes in E. coli that have been studied for, seems like 1,000 years. In the lac operon, there's another sequence here before -35 that binds another protein that's also involved in regulation. So there are other things that can be happening, other sites that can bind other proteins that can be involved in assisting in RNA polymerase binding or having a negative effect on RNA polymerase binding. And so there's more going on in terms of regulation of the promoter here than just whether the RNA polymerase binds. There are other protein factors that can bind to the promoter and influence transcription. It's just we don't need to worry about those for the purpose of this course right now. Okay? Yes?
I'm just thinking about like diseases. So do we really need the whole sequence for, like, for example, -35 box and -10 box for the polymerase to bind, or if I have a mutation in one of them, it would still work?
So let's suppose that this gene is a highly expressed gene, binds RNA polymerase really efficiently, gets a high level of expression. If a mutation occurs here that changes this AT pair to a GC pair, that's almost certainly going to decrease binding of RNA polymerase and decrease expression. If we're talking about E. coli, the E. coli may still be able to survive, but they may just grow more slowly,
Okay.
less efficiently.
Yeah.
There are some mutations that can be deadly and just shut down transcription completely, and then the organism might die. So some mutations can be lethal, some can just decrease the fitness of the organism. Okay? Yeah.
Would that be if the mutation caused something like a stop codon, that would then cause a catastrophic mutation?
So if there was so the start codon is over here and stop down there. If there was a mutation that caused the stop codon now to appear in the middle of the gene, then what that's going to mean is your protein's going to start translating here. It will stop here. You'll make a truncated protein. That protein will not be functional, and that may or may not be lethal to the cell. Right. Yeah.
So you're saying doctor, that has to be within the ORF for it to be read and translated into a catastrophic failure or whatever?
So from here to here is referred to, as you just said, the open reading frame. Write that down. That's abbreviated ORF. So ORF, or open reading frame. The open reading frame goes from the start codon to the stop codon, and that just means that in this entire coding region here, there are no stop codons. That's an open reading frame, no stop codon. If a stop codon does appear somewhere in this region, that's almost assuredly going to be a mutation that prevents the gene from functioning, prevents the protein from functioning, may be lethal to the cell. It's possible you could have a mutation like that in the promoter that prevents transcription, and that would be equally bad, right? So it doesn't have to just be in the ORF, it could be in the regulatory region as well. If it's a mutation in the regulatory region that prevents transcription, then that's just as bad as having a mutation in the coding sequence. Okay, does that make sense? Okay, cool. All right, that's bacteria. How do I have? Let's start with eukaryotes. I don't know that I'll finish, but we'll start. And we're going to contrast what happens in bacteria to what happens in eukaryotes. So the first thing to remember is that in eukaryotes, transcription is happening in the nucleus, where the chromosomes are. RNA processing happens in the nucleus, so the conversion of the pre-mRNA to the mRNA happens in the nucleus. Only when the RNA has been processed and is now a final mRNA does it then depart the nucleus and go to the cytoplasm, where it's going to get translated by a ribosome. Okay? So the transcription and translation processes in eukaryotes are completely separate. If we're talking about nucleogenes, right? If we're talking about the mitochondria, then it happens like in bacteria, right? Some of you may have heard of what's called the endosymbiont hypothesis. Not really much of a hypothesis anymore. People are pretty certain that there's a lot of evidence to support this. So the idea of the endosymbiont hypothesis is that mitochondria used to be bacteria, and some big pre-eukaryotic cell engulfed a smaller bacterium, alpha proteobacterium, and engulfed it, and then that cell that got engulfed, the smaller cell, evolved over time into the mitochondria. And because of that, mitochondria superficially look like bacteria. Their genomes look like bacterial genomes. They're circular. Their genes are organized like bacteria. They get transcribed and translated like bacteria. It's coupled like in bacteria. There's a lot of biochemistry going on that's like bacteria. So, and then if you look at the DNA sequences of mitochondrial genomes, they look like alpha proteobacterial genomes. So there's a lot of support for this idea that mitochondria started out as an endosymbiont. And the same is true for chloroplast, same kind of evidence there. Okay, so a little aside. Oh, and the endosymbiont hypothesis was proposed by Lynn Margulis. She has since passed away a number of years ago, but I have to give her a shout out because she was at UMass Amherst, just across the river. When she first proposed this hypothesis, I thought she was off her rocker, but she was absolutely right. Absolutely right. Okay, so anyway. All right, so just keep in mind that we've got transcription and translation uncoupled here. The first thing I want to tell you is that this promoter region, so this region here in front of +1, this is the promoter, and you can see in the promoter now I have multiple boxes. Here's a promoter that's got 1, 2, 3, 4, 5 boxes. These are 5 different binding sites for different proteins. Promoters in eukaryotes are much more complicated. They have lots of different boxes or motifs for binding different proteins. Those proteins in eukaryotes are referred to as transcription factors. And no joke, a eukaryotic promoter can bind 30, 40, 50, 60, 70 transcription factors. So it's very complicated, very complicated. So what I want you to imagine is that we've got a region of DNA that's our promoter in a eukaryote, so I'll just draw the promoter as one big box here. And you have to imagine that on this box, or on this promoter region, we're binding different transcription factors. And then there are transcription factors that bind to the transcription factors, and so on. So it's really complicated. Another difference in eukaryotes, in addition to this complexity, is that the RNA polymerase in eukaryotes is completely incapable of binding the promoter on its own. That's not true in bacteria. But in eukaryotes, you need these transcription factors on there, and only then can the RNA polymerase come on and bind. Okay? How well the RNA polymerase binds kind of depends on how many transcription factors are there. It's like the more the merrier. The more transcription factors on here, the better the RNA polymerase binds. And so this arrangement is really complicated, but it means we can really fine-tune our expression. You want to dial up expression a little bit, add a few more transcription factors. You want to dial it down, subtract a few transcription factors, right? So transcription in eukaryotes can be really fine-tuned. Whereas in bacteria, a lot of regulation is just on-off. In eukaryotes, it's not just on-off. It's like think about a meter. It's like you can really dial things up or down. The All-Star game for baseball was last night, so of course I was thinking about transcription factors while I was watching the game after class last night. And you kind of think of, so those of you who know baseball, the catcher who catches the pitches from the pitcher has a big mitt. It's a mitt that's bigger than any other player has on the field, right? And kind of the bigger the mitt, the easier it is for the catcher to, engulf the ball, right? And it's kind of the same here. You can think of this as sort of a big catcher's mitt that's trying to catch that RNA polymerase. And so, it's maybe not the best analogy, but there you go. So it's complicated. A lot of transcription factor binding sites, a lot of transcription factors, and then transcription factors that bind to transcription factors. So there are well-known examples where you get a transcription factor bound to the DNA, and then another transcription factor binds to that, and then two more bind to that, and another one binds to that. So it's really complicated. How many genes in eukaryotes do we thoroughly understand the regulation of? Not very many, because it's really complicated. In bacteria, we understand the regulation of lots of genes really well, but in eukaryotes, it's just really complicated. Okay. So, what I want you to imagine is transcription factors bound to these different boxes, and then transcription factors bound to the transcription factors, and transcription factors bound to those. It's worse than that, because many eukaryotic genes, extend this out here, so now we're way up here. Many eukaryotic genes have another box out here, that I'm going to label E, and that thing is an enhancer. Enhancers also bind transcription factors, and so you can think of this enhancer as forming an additional blob of transcription factors. Now, these enhancers can be pretty far away. They can be upstream of the gene. They can actually be downstream of the gene. Down here somewhere, maybe like a block that direction, or 2 blocks in that direction. They can be thousands of base pairs away, up or downstream. but they're binding transcription factors, and they help transcription. And how on earth do they help transcription if they're so far away? And the answer is that they're far enough away that the DNA can bend around, and the enhancer, with all of its transcription factors, can bend around and come in contact with the promoter with all of its transcription factors and help bind the RNA polymerase. So you've got transcription factors here, you've got transcription factors there. This thing bends around, contacts the transcription factors here, and makes a bigger tetrasmith, right, for grabbing the RNA polymerase. The enhancer and the promoter are not contacting one another directly. It's the transcription factors bound here binding to the transcription factors bound here. Ooh, what a nightmare. So, with all that going on, imagine trying to understand how regulation is going on in detail for any given eukaryotic gene. It's really complicated, really complicated. In eukaryotes, this means we have a lot of genes that code for transcription factors. Transcription factors are proteins that are involved in gene regulation, and we have a lot of genes coding for a lot of different transcription factors. Why are our different cell types different? It's because in brain cells, we're producing different transcription factors than we are in liver cells. So the brain cells are expressing different genes than the liver cells. The brain cells are expressing some of the same genes as liver cells, but at different levels because of the transcription factors being produced. Pretty cool. Complicated, but cool. I always tell my students that if you like order and you like being able to really figure things out and understand things down to the nth detail, right? Work on prokaryotes, work on bacteria. If you like complexity and confusing situations and really complicated puzzles, then work on gene expression in eukaryotes because it's a nightmare, right? So, yeah, anyway. So, I'm just trying to give you a flavor of how different things are. And from this, I think you can see quite obviously why if we have a eukaryotic gene with the eukaryotic promoter and we put that into bacteria, bacteria doesn't know what to do with this eukaryotic promoter. A eukaryotic promoter doesn't look anything like a bacterial promoter. So that's why we have to take our eukaryotic gene and when we put it in bacteria, give it a bacterial promoter, because this thing is just not going to work in bacteria at all. Yeah.
Are there any theories about the evolutionary advantage of all that complexity?
Yes. The idea is that prokaryotes have taken the strategy, mostly single cells, grow really efficiently, reduce complexity so that they can grow really fast. They don't have to make so many proteins, and there's not so much DNA that has to be replicated every cell cycle. Eukaryotes have gone completely in the other direction. Really complicated big genomes, lots of DNA, lots of different proteins, really complicated regulation. But the advantage is that you can do this really fine-tuning of the gene expression. So, two different strategies that both obviously seem to work. We're here after all, so even though we're big and clunky and complicated, we still manage to eke out an existence, right?
So the enhancer is always located downstream of the promoter?
No, the enhancer can be located up here in front of the gene. It can be located out here downstream of the gene. There are even examples of enhancers being located in an intron. Yeah.
So in eukaryotes, the transcription factors are the things that primarily control how much a gene is expressed.
Right.
In prokaryotes, is it only the sequence of the -10 and the -35 box that controls that?
Not only the sequence, because there are, for some genes, it's only the -10 and 35, but for a lot of genes, there are additional binding sites here that bind additional proteins. It's not anything like that. It's nothing that complicated. It's one or two proteins that bind that either assist or reduce transcription. So it's not just the -10 and -35. For some genes it is actually, yeah.
You said like the enhancer and the promoter never really interact with each other directly, but through the transcription
Through the
factors.
transcription
Just
factors,
thinking
right.
about it visually, you know, if it's downstream very far away from the promoter, do they like build bridges, or like how do they directly, like how do the transcription factors interact with each other?
So.
Like just distance
Yeah, so
wise,
the
like-
understanding is that the way these transcription factors interact with the transcription factors down here is the DNA has to bend around
Okay,
and
yeah, that makes sense.
so that this thing can come in
Yeah.
contact, right? That's why the enhancer can't be too close, because if it's too close, it can't bend around to make the contact. So if you take that enhancer and move it a little farther away, that'll be okay. If you move it a little closer, that'll be okay. But if you move it too close, then it doesn't work anymore, because you can't get the appropriate bending around for the transcription factors to interact appropriately. The enhancer can be thousands of base pairs away, but for some reason that we don't completely understand, if it gets too far away, then it doesn't work anymore. Yes.
So that would be based on chemical affinity, based on protein domain pockets.
Yep, so it's just, interactions of different types between the proteins, right? Sir? Yeah, so, so the famous lac operon is an example where there's, there are actually two additional sites that combine two different proteins that are involved in regulation. One is involved in inhibiting expression, and the other one's involved in accelerating expression. So there's both positive and negative regulation there, but we're not gonna do that. This was a genetics class, we do that, but we don't have to do that here. Any other questions about what I've talked about so far? I'm not done yet, obviously. So there are other things that are more complicated in eukaryotes. Another complication is that the eukaryotic transcript doesn't really have a ribosome binding site. In bacteria, we have a ribosome binding site that told the ribosome where to bind and where to start translation. We don't really have that in eukaryotes. In eukaryotes, what happens is that this pre-mRNA gets processed to the final mRNA. So we're going to get our RNA processing. And in that RNA processing, the 5' end of our transcript gets that cap, right? Cap. That cap structure is important for binding the ribosome. And so in eukaryotes, the ribosome actually binds, again, it's the small subunit of the ribosome, binds actually at the 5' end of the transcript. But the start codon, you know, it looks short here, but the start codon can actually be thousands of nucleotides away. And in that region, there can be multiple AUGs. So how does the ribosome know what's the right AUG? If there's an AUG that's close and one that's farther away, and then here's the real one that's supposed to be the start codon, how on earth does the ribosome, which binds down here, know that that third AUG is the one I'm supposed to use and not the first or second one, right? Part of the answer has to do with something called a Kozak sequence. And the Kozak sequence is a sequence around the start codon. And that Kozak sequence looks like this. GCC, although I've put it in brackets because that's not as conserved across different genes as these other nucleotides are. Then an R. An R means a purine, which is either A or G, right? So R means A or G. Then CC, then the start codon AUG, and then a G. And And that sequence tells the ribosome, oh, I'm the right one, pick me, pick me. And so that is an important feature in some genes. But it turns out not all genes have a Kozak sequence. And so what's going on in those genes? How does the ribosome know where the right place to start is? And that's still a bit of a mystery. It looks like one thing that's important is how the RNA folds three-dimensionally. That looks like that's important. But really, I can't tell you with any assurance exactly what's going on for all genes. This is certainly how it works for some genes with this Kozak sequence. So if you look at mouse and human genes, you'll see that some have a Kozak sequence and some don't. So don't think of it as any kind of a rule. Okay, so how translation knows exactly where to begin, what start codon to use, is a little bit of a mystery, and still being worked on, and appears to be different for different genes. The other thing I want to talk about is splicing out of the introns. So I've just shown exon 1 here, and then one intron, and then the start of exon 2, and this thing may have 50 introns and 50 exons, but we're just looking at the beginning of the gene here. There's something called the GUAG rule, and that is at the start of the intron, the first two nucleotides are almost always GU. Almost always GU. And the last two nucleotides are almost always AG. So that's the GUAG rule. And the cut is made for splicing right in front of the G and right after this G, right? But two nucleotides is certainly not sufficient to be specific, right? You're going to find lots of GUs in here. You're going to find lots of AGs. So how, again, does the splicing machinery know that this is the right place to cut here and this is the right place to cut here? And again, the answer seems to be not just the GUAG, but also how that RNA folds up seems to be important. And the splicing is done by a really complicated molecular machine called a spliceosome. So the spliceosome does the splicing. It's a big complicated molecular machine analogous to a ribosome. It's made up of about half protein and half RNA like a ribosome is. And this big complicated molecular machine somehow knows how to do the splicing. And it's got to be perfect because, remember, we're reading in triplets, right? We're reading in triplets. And then we splice out this region here and we keep reading in triplets. If there's a one nucleotide error, that's going to completely throw off that triplet reading frame, and then the protein will be garbage that's made. So the splicing has to be absolutely perfect, down to the nucleotide, right? A one nucleotide error, disaster, right? Now I want you to think about this. Do you think errors are ever made?
Yeah.
Yes.
Of course. Nothing is perfect in biochemistry. I always tell my students nothing is 100% in biochemistry. There's going to be an error rate every time you do a splicing event. Now think about these genes that have 100 introns and 100 exons. If you've got 100 introns, you're going to have 200 splicing events, right? Because for every intron, you got to splice twice, right? Do you think you're making some protein that's bad from that? Certainly. And the more introns you have, the more error you're going to have and the more faulty protein you're going to make. Sounds like a recipe for disaster, right? But again, we're still here and we're still working. But that's a puzzle because it seems like having a lot of introns and a lot of exons would be selected against evolutionarily. But there must be some advantage to it because we have these genes that are just loaded with introns and exons, right? So just think about that a little bit. And if you can come up with a really good answer that can be proven scientifically, you're next in line for the Nobel Prize. Okay? Yeah.
It's a frame shift, right?
If you delete, if you missplice by one here, then you're going to have a frame shift mutation, which is going to throw off that reading frame, and you're gonna make, from the point of the frame shift, from there on, your protein will be garbage. Yes?
So you talked about like RNAs, mRNAs. What about like microRNAs?
So microRNAs are short RNAs that are found in eukaryotes. They're coded for by genes, microRNA genes. These are small genes, and those microRNA genes code for RNAs called microRNAs that are functional in regulation. So microRNAs are not translated, but they work in regulation. And I'll talk about those later on.
And then for this, because of the building of it, is there a separate process for like the degradation of it, like the breakdown of it?
There are processes for degrading RNAs. That's why they have a half-life. So I mentioned that in bacteria, the half-life is pretty short, 3 to 4 minutes for transcripts. In eukaryotes, much longer. How long the half-life is for an RNA in both bacteria and in eukaryotes depends partly on the three-dimensional structure, again, that RNA folds up into. Kind of the more folded up it is, the longer its half-life is going to be because it's a more compact structure. If it's more splayed out, then it's easier for ribonucleases to chew on it and degrade it, and then it's going to have a shorter half-life. That's sort of a hand-wavy answer, but there are enzymes inside both bacterial cells and eukaryotic cells that degrade RNA. And so no RNA is going to last forever. Some of the really stable RNAs are like the ribosomal RNAs found in ribosomes. They're really stable because they're packaged inside a ribosome and they're protected by a lot of proteins that are bound to the ribosomal RNA. And so those RNAs are really stable. Yeah.
So there can be transcription errors, splicing errors, and translational errors, and they can all propagate. So, I mean, how is it not just a total disaster? What is like the error handling that the cell actually does?
So, when mistakes like that are made, faulty protein is made, and those proteins get degraded pretty quickly. So we have mechanisms in our cells for degrading proteins that are not folded properly, and that doesn't completely answer the question. We have mechanisms for dealing with those bad proteins. Once a transcript is made, if that transcript is faulty, it's going to continue to encode bad protein, right? And we don't really have a good mechanism for getting rid of faulty RNAs, because think about if the RNA polymerase makes a single mistake in that RNA, which changes a single nucleotide, which changes a single amino acid, which makes that protein non-functional, the cell has no way of recognizing that RNA that's maybe 1,200 nucleotides long has a single mistake in it, right? And so that RNA is going to persist and keep getting translated into bad protein, which then needs to be gotten rid of. And so all these mistakes, the cell's paying a price for, but we must make enough good protein that we're still here. So one of the things that really bothers me in eukaryotic molecular biology is this issue of really big genes with lots of introns and exons, because the error rate there is very high, and I don't understand why that hasn't been selected against, right? A gene that has 100 introns and 100 exons is just ridiculous, right? So I've yet to see an explanation that makes that clear. And somebody will come up with a reasonable explanation sometime, but I just haven't seen it.
Isn't that just an evolutionary advantage of the chance of mutation?
Well, so the thing about introns and exons, and I gotta watch the clock here. The thing about introns and exons is that there is an advantage. We haven't gotten to this yet. There is an advantage to having multiple introns and exons, because you can actually make different forms of the protein from the same gene on how you splice your RNA. So that's called alternative splicing, and that's cool, and I'll show it to you later. But a lot of genes that have 50 introns and 50 exons, some of those genes aren't doing very much alternative splicing. So that can't be the full answer. So there's still a lot of mystery to it. And again, if one of you can solve this, then I'll nominate you for the Nobel Prize. Okay? Anyway, all right, we need to move on to protocols. But I hope that what I've done here is to show you how different things are and why when we clone a eukaryotic gene into a bacterium, or the other way around, clone a bacterial gene into a eukaryotic cell, why we have to worry about all this stuff. We have to give it a promoter that works in that host cell. We have to, if we're cloning a eukaryotic gene into a prokaryotic cell, into a bacterial cell, we have to get rid of the introns, right? Fortunately, we can get rid of the introns by just using cDNA. That's pretty cool. So that's a neat thing. But we do have to worry about all these issues. Otherwise, our experiments won't work. In the very early days of molecular biology, people took eukaryotic genes and put them in bacterial cells and thought, oh, this ought to work. Nothing. Nothing. A lot of wasted years went into experiments like that. Okay. Now we're smarter and we know why. All right, let's go to the protocols, and we're dealing with protocols from 3 separate experiments here. Ooh, my screen's doing something silly. Give me just a second, I have to actually log in to use this. Sorry, there's something screwy happening with the system here. Talk amongst yourselves for a minute.
Did we get our RT-PCR samples back yesterday?
Which
So, sorry, experiment 3.
Yeah, we put them. We put the samples somewhere. We did something with it. Oh, wasn't that no?
yesterday we just set up the PCR.
Set up the PCR.
Today they're gonna
We
be
did, but then oh no, he said we're gonna get them back today.
We're getting them back there, okay.
Yeah, that's
Oh, okay, so
Yeah.
we're gonna get them back today.
Yeah, so that's just a little confusing. What did we do? Why? Well,
I know,
this part was
the multiple
silent.
experiments, it's hard to
Yeah.
keep track. So yeah, we're doing the we're trying to isolate the TTR gene because it's abundant in the liver. We've amplified it. That's it. There's no vectors or PMALs or anything in this experiment. All right, I'm caught up. Are you able to switch between the experiments in your head pretty quickly? It always takes me a second.
If I like, if you set them up
The screen
yourself,
on my
yes,
computer is
because
completely
this is
lost
not
its
like
mind. It's showing
I
up
don't know.
here just
Everyone
fine,
thinks differently,
fortunately.
right? So
But what I'm looking at here
Yeah.
is really bad, so bear with me a second here. You know what, I'm gonna raise this one so we can.
because like in the lab, I do like several things at the same time as well. But because I've set them up and I know what I'm doing.
Right,
Yes.
right.
but
So the
yeah,
order in which I'm gonna go through the 3 events here is not necessarily the same order you're gonna do downstairs, right? When you get downstairs, do it in the order they tell you. I'm gonna go through it in this order, but I do not believe that's the order you're gonna do it in downstairs. But I'm gonna start with experiment 3, and it's loading our RNA drill. And this is going to be on page. 113.
So we just wanna see that it's actually there.
So I want you to remember that yesterday you did your RT-PCR reaction, and that was on pages 109 to 113. And previously, back what seems like 2 weeks ago now, you isolated RNA from mouse liver tissue, and remember you divided up all those aliquots into different tubes to use in experiment 1, experiment 4, and experiment 3. 2 of those aliquots were labeled RNA liver for gel 10 microliters and RNA liver for gel 5 microliters. We're finally going to run your RNA on a gel to look at it. Now, normally we would have done this right after you made your RNA, but we didn't have time. We had to just keep forging ahead with the experiments. You certainly had good quality RNA according to the nanospec. You had a nice large amount of RNA according to the qubit, but we haven't looked at it on a gel yet. So I'm here to tell you that in your own labs, you would want to look at it on a gel before proceeding. We just didn't have time to do that. So now today we finally have time, so now we're going to put that RNA on the gel. We're also going to put on your RNA from your, or your DNA rather, from your RT-PCR reaction. So this is on page 113. Run mouse liver RNA and RT-PCR reaction on an agarose gel. I want you to notice that this may seem kind of weird to you. We're running RNA on this gel and we're running DNA on the gel. And that's okay, it works just fine. A lot of people would raise their eyebrows and go, you're doing what? But you'll see that it works just fine. In step 1, the overzealous staff has already poured your 1.5% agarose gel. Again, we're not always going to do this for you, but this is to get things moving along this morning. In step 2, remove a 10 microliter aliquot of your RT-PCR tube and place the sample into a new green 1.7 ml microtube tube. Label this tube RT-PCR gel. Add 2 microliters of PD or purple dye to the tube and mix. Save the remainder of your RT-PCR tube in your -20 box. This is in case we need it later. This RT-PCR tube is the product of your RT-PCR reaction from page 111, your RT-PCR reaction we ran late yesterday. Everybody remember that?
Yeah.
Good, you guys are doing good. Okay. In step 3, the 2 total RNA samples that you prepared for the gel on day 2, steps 15, 16 on page 106, if you want to go back and look at that, will be returned to you in your ice bucket. These have been sitting in the -80 because they're RNA. Remember, we collected all those tubes from you and put them in -80. These are labeled RNA liver for gel 10 microliters and RNA liver for gel 5 microliters. Add 5 microliters of water to the RNA liver for gel 5 microliter tube. Only to the 5 microliter tube. Now both of your tubes have 10 microliters of solution, right? Everybody clear on that? Good? Okay. You're just making it so both tubes have 10 microliters of solution. Then pipette up and down the mix. Both tubes now contain 10 microliters. In step 4, add 2 microliters of purple dye to each tube and stir to mix. And now they both have 12 microliters, right? In step 5, denature both of the RNA samples from step 3 above. Actually, it's from step 4 above because you added the purple dye. By heating them to 65 degrees for 3 minutes. Now, the reason for heating these RNA samples is we want the RNA to run on the gel according to its length, right? But since RNA is single-stranded, it likes to fold up on itself, right? So in order to get the RNA to run according to its true molecular weight, we need to heat that RNA to open it up and then load it on the gel, okay? The refolding takes time, so we're going to heat it, we're going to put it on ice, we're going to load it on the gel, okay? In step 5, you're going to denature both your samples, 65 degrees for 3 minutes, spin briefly in your nanotube to get all the liquid to the bottom. Place on ice immediately for 1 minute. This will slow down the refolding of the RNA. 1 minute or until you're ready to load your sample on the gel. So basically, put your sample on ice and leave it there until you load it on the gel. In step 6, load the agarose gel with samples in the following order. Note you're going to skip lane 3. Remember, your lanes are across the top, right? 1, 2, 3, 4, 5. Okay? Lane 1, 12 microliters of RNA liver for gel, 10 microliters. So this is a total of 12 microliters now because we added 2 microliters of the purple dye. In number 2, 12 microliters of RNA liver for gel, 5 microliters. It's also 12 microliters because we added 5 microliters of water and 2 microliters of purple dye, right? Your lane 2 should have half the RNA as in lane 1, right? And the reason we're doing this is that sometimes lane 1 will have too much RNA and it won't give us a very good view of our RNA, so then we run another lane that's got a little less RNA. Okay, so that's why we're running 2 different amounts. And you'll see that when we look at our gels later. Then we're going to skip lane 3, so lane 3 will be blank. Lanes 1 and 2, remember, have RNA. Okay. Then in lane 4, we're going to run 10 microliters of 100 base pair ladder in purple dye. So you've used this before. It's the one that gives us a band at 100, 200, 300, 400, 500. This should be in your 4-degree box, right? Lane 5 will be 12 microliters from your RT-PCR gel tube that you prepared in step 2. So this is your RT-PCR reaction where we're asking the question, is the TTR gene expressed in mouse liver, right? Just yes or no. We see a band, it's yes. We see a band at 150 base pairs, it's yes. We don't see a band, it's no. We see a band at 1100 base pairs, which is the genomic size, and no band at 150, then the answer is still no, right? We see a band at 1100 and a band at 150, then the answer is yes. So we're looking for that band at 150 base pairs, which remember is the size that you get if it's amplified off of messenger RNA. Review your notes from last night for that. Okay, that's lane 5. Now, the rest of the lanes are going to be the controls that the staff prepared, and these will be delivered to you in green tubes on ice. Lane 6, we want you to load 15 microliters of the DNA contamination control. This is the control we ran where we purposefully spiked with genomic DNA. So you can see what happens if you have genomic DNA contaminating your RNA sample. Lane 7 is the no RT control. Doesn't have any enzymes in it. So it should give nothing, right? No enzymes, no product, right? Lane 8 is the no template control. If you don't put any template in, then you should also get no product, right? So we'll talk about these controls again when we get to gel results. Then lane 9 will be another 100 base pair ladder marker, 10 microliters, and then lane 10 will be blank, okay? In step 8, run the gel at 50 volts at room temperature for about 2 hours. The purple dye should be about 2/3 of the way down the gel before stopping the electrophoresis. The staff will watch these and will turn off the gel boxes at the appropriate time if you're not in lab. And so that's load the RNA gel. That's the first thing you're doing. Well, maybe not the first thing downstairs. They'll tell you the order. Okay, purification of gap pH and PMOL PCR product in experiment 1. Put this up for you, and I didn't even go to the right place. Anyway, you guys were all looking at your manuals, you didn't notice, right?
No.
Okay, good. Alright, so we are now going to experiment 1.
What is it, day 3? No, day 4.
Day 4. So experiment 1
Day
now.
4. 31.
So, on page 30, you will recall that last night you put 2 to 3 microliters micrograms of your GAP-DH PCR product into a tube with the SPF1 restriction enzyme. And remember, the idea here was to cut your GAP-DH PCR product at the SPF1 site to generate a sticky end at the distal end of our cDNA. And remember, this is to facilitate that unidirectional cloning into the vector. So we cut the GAP-DH with the restriction enzyme, SPF1, and then we also cut the PMAL cloning vector with two enzymes, XMN1, which would give a blunt end on one side, and the SBF to give a sticky end on the other side. And so you have these two reactions, and we let those digest overnight. And now what we have to do is we have to inactivate those restriction enzymes, and then we're going to purify these digests to get rid of the enzymes, okay? So we're first going to inactivate and then purify. So the purpose of this part of the protocol is to heat and activate the restriction enzymes and then purify your digest by removing all the enzymes. And we also want to get rid of the short DNA molecules. So when you cut your cDNA, you cut off this little bit of DNA at the end here, so there's that little fragment. Remember, when you cut the vector, you cut the plasmid in two places and release about a 40 base pair little fragment. So we want to get rid of those, so we'll do that in this cleanup also. So to purify your restriction digest in the second paragraph here, you will again be using the NEB Monarch DNA cleanup kit. It's a silica-based system. We used the same cleanup kit yesterday. This is again using that salt grid system. It'll bind our big DNA, but it will not bind these little tiny fragments. The little tiny fragments don't have enough negative charge to bind to the resin in the column. So in the column, at a pH less than or equal to 7.5, the long double-stranded PCR product and the plasmid in the other digest will bind to the silica, while the short fragments and the restriction enzymes will not bind. So that means we're going to be able to wash these out, and we'll be left with just our DNA that we want on the column. So in step 1, incubate the restriction digest at 80 degrees for 20 minutes to denature and inactivate the restriction enzymes. This will be done by the Notorious staff while you're in lecture. So they're doing that right now. In step 2, to each of the two 1.7 ml restriction digest tubes, one's labeled GAP-DH PCR SBF, and the other one's labeled PMAL XMN. These each contain 50 microliters of digest, because the two digests reach 50 microliters. To each of those, add 250 microliters, or 5 volumes of the binding buffer for this protocol, and mix by pipetting up and down. Again, you want to wear gloves because the binding buffer contains guanidine isothiocyanate, which is a cyanide derivative, so you want to be wearing your gloves all the time anyway to protect your DNA, so that's kind of a no-brainer.
I was just wondering, why do you actually have to heat and activate when you can just wash them out?
Yeah, so why do we heat and activate and then wash them out? Why not just wash them out? And the answer is that restriction enzymes like to really bind DNA.
Oh, okay.
So even after they've cut the DNA, the restriction enzymes tend to hang on to one of the two fragments. And so by heating, we bump the restriction enzyme off the DNA, and then it's easier to get rid of them. Okay? Good question. That was step 2. In step 3, you will receive 2 columns and 2 collection tubes. Insert each column into a collection tube. Label one column and one collection tube GAP-DH. Label the other column and the other collection tube PMAL. Apply each 300 microliter sample to the appropriate spin column and the appropriate collection tube. If you happen to put the wrong one into the wrong tube, then just change your label on the tube, right? But try to get the right thing into the right tube so we can keep things simple here. In this step, the large PCR product in one tube and the plasmid in the other tube will bind to the silica spin columns. Centrifuge the 2 columns for 1 minute at 10,000 times G in the microfuge. Discard the flow through from each collection tube because the DNA you want is bound to the column. Then put the spin column back into the same collection tube. Now we're gonna do the washes where we wash out the material we don't want. So in step 7, add 200 microliters of DNA wash buffer to each column. This buffer contains ethanol and the buffer and is used to wash the columns. Centrifuge the spin columns again for a minute at 10,000 times G in a microfuge. Discard the flow through, which is the wash buffer with stuff that we don't want. Place the spin columns back into the same 2 ml collection tubes. Again, add 200 microliters of wash buffer to each column, and again centrifuge for a minute at 10,000 in microfuge, again to remove the wash buffer and the material we don't want. In step 11, transfer the gap pH column into a 1.7 ml blue tube and label it pure gap pH. Transfer the PMOL column to a 1.7 ml blue tube and label it pure PMOL. Be sure the tip of the column doesn't contact the flow through, as we said again. If you do that, please contact the staff and they'll tell you what to do. In step 12, to elute the DNA from the columns, add 30 microliters of DNA elution buffer to the top center of each column. The DNA will elute from the columns in low salt at pH 8.5. And again, when you add this 30 microliters, same thing I told you yesterday, if we're looking down into the microfuge tube like this, so the cap will be over here on, or the hinge will be over here on one side. We're looking down into the tube, down at the matrix. You want to pipette your liquid into the center, okay? It's only 30 microliters, and we want it to sort of spread out evenly and then wash through and get your DNA out of the column. If you pipette it off to the side, you're not going to get all of your DNA back. Okay. In step 13, let the column stand for a minute at room temperature. That's important actually. It starts to seep into the column and helps your DNA to release. And then centrifuge for a minute at 10,000 times G in the microfuge to elute the purified DNA. Discard the columns, close the lids, and store the tubes in your 4-degree box until tomorrow. So we're purifying your PCR product and your plasmid, and then we're just going to put it away until tomorrow because we have other stuff that we got to get done today. So we'll come back to those tomorrow and continue on with experiment 1. That's going to be it for experiment 1 today. Finally, the last thing we have to do has to do with our DPN digest in experiment 5. So orange shirt day, right? A lot of experiment 5 going on. If you go to page 154 in your manual. Remember that we have these 2 versions of our plasmid that we inserted that 20 nucleotide sequence into. So here's the 20 nucleotides in green. Remember, this is from that inverse PCR that we did. And this is the plasmid PCAS that has the Cas9 gene on it. This is the one we're going to use for CRISPR later on. These molecules are the ones that were generated by the inverse PCR, and they were not cut by DPN1 because they were not methylated. But we cut with DPN1 last night, and it would cut these original molecules, which don't have our insert, and it would cut them up into small fragments. And it's these small fragments that we now need to get rid of today, and that's what we're going to do in this next protocol. So on page 155, late yesterday you cut with DPN1 and we let it digest overnight. This is the reaction you set up. It was a 25 microliter reaction. been cutting at 37 degrees overnight. And now today we need to purify and get rid of those fragments from the cut up plasmid that we don't want. So day 4 purify and side select the DPN1 digested PCAS adenine 2 plasmid. So the purpose of this protocol is to remove the proteins, that is the restriction enzymes, the excess primers that were in there because of the PCR that we ran, and the linear digested DNA fragments from the DPN1 digest of the plasmid molecules that we don't want. So to do this, we're going to use chromospin 400 columns. These come from the company Takara. These are gel filtration columns that exclude molecules larger than the pore size from the resin. So these are gel exclusion columns. So this is new to us, something we haven't done yet. Don't want you to worry about the time. You'll notice that we have this little 11:30 to 12 lecture here that we may or may not have. So if lab goes long and we're pushing 12 o'clock, don't worry about it. Okay. So these gel exclusion columns work like this. So the columns have these beads in them. Kind of looks like this. Obviously an oversimplification, but this is the column material, has these beads. And what happens is that when you load on your sample on here, small molecules like the digested plasmid DNA, those small molecules can actually penetrate the beads and they'll get stuck in the column. They'll get stuck in the column. The big molecules, which are the big plasmids we have that aren't cut, those molecules are so big that they can't penetrate the beads. They can't go in. So what do? They just go around the beads, traverse through the column, and they come right out really fast. So it's a little bit counterintuitive. The big ones are going to come right through, and the small ones get hung up in the column. Okay, so that's the strategy that we're using here with these chromospin columns, and these chromospin columns work great. It's a really good trick to know. So, because the next step in this project is a transformation step, we're going to replace the column storage buffer with 1X-STE buffer, which is just TE, which is tris-EDTA, with some sodium chloride. And this will keep our circular plasmids stable. It'll keep them intact when we do the transformation. And when we do the transformation, that is when we put this plasmid into the yeast cells. I'll talk to you a little bit more about that. So we'll come back to that. All right, in step 1, each group will receive their 30 microliter DPN1 digest tube containing their PCAS adenine 2 plasmid. Raise the volume of this sample to 70 microliters by adding 40 microliters of the 1X-STE buffer to the sample. In step 2, each group will receive a chromospin 400 column containing 1X-TE buffer, labeled the column PCAS adenine 2. Again, we're calling this PCAS adenine 2 because it's got that 20 nucleotide adenine 2 targeting sequence on it. It doesn't have the whole adenine 2 gene, it's only got 20 nucleotides on it. Okay? In step 3, vigorously shake the chromospin column several times to completely resuspend the gel matrix in the storage buffer. These are gel beads, so that's what we're talking about in terms of this matrix. In step 4, remove the top cap first and then the bottom cap from the chromospin column. Save the top cap and dispose of the bottom cap. The bottom cap is not really a cap. It's one of these sort of twist-off caps. So when you twist that off, it's going to break off and then you just throw it away. So that's what I'm referring to as the bottom cap here. Place the bottom tip of the column gently into one of the 2 ml collection tubes provided. In step 5, discard any buffer that is collected in the 2 ml collection tube because some of the buffer from the column will just drain right through. Replace the column gently into the same microfuge tube, then place the entire assembly into a 15 ml conical tube and label the cap at the 15 mil tube with your group number. So you're gonna have this chroma spin assembly dropped into a 15 mil tube so that we can spin it in the centrifuge. In step 6, centrifuge the column in a swinging bucket rotor or in a fixed angle rotor at 700 times G for 3 minutes to remove the storage buffer. So this is just to get out the buffer that's already in the column. In step 7, remove the column assembly from the 15 ml tube with tweezers because you can't get your fingers down in there. Discard the collected buffer from the 2 mil tube into a waste beaker. In step 8, there's a different kind of cap now that we're going to put on the bottom. So put the new bottom cap onto the column. Staff will show you all this. Add 1 mil of 1X TE, 1X STE to the column, and replace the top cap onto the column. Resuspend the matrix completely by inverting the column several times. And then in step 9, remove the top cap and then the bottom cap from the column and put it back into the 2 mil collection tube. Now put this whole assembly into the 15 mil collection tube and centrifuge at 700 times G for 3 minutes. Remember that the DNA you want is going to come right through. So unlike in some previous protocols, you're not going to throw away the flow through here, right? Okay. Then you're going to repeat steps 7, 8, and 9. Oh, sorry. We're just washing through buffer at this point. So you are going to throw it away. So we're just washing the column. Sorry, we haven't put your DNA on that. So we're going to repeat the wash in 7, 8, and 9. Then in step 11, remove the column assembly from the 15 ml tube with tweezers. Discard the collected buffer because it was just wash buffer. From the 2 ml tube into a waste beaker, assemble the column again using the same collection tube into the 15 ml tube, and centrifuge now for 3 minutes to remove the residual buffer. So now we're just getting rid of last traces of buffer that are in your column. Okay. Now we're ready to add the DNA. So in step 12, remove the column assembly from the 15 ml tube again. Place the wash spin column into a new second microfuge tube, because now we want to be using a clean tube, because this is where your DNA is going to go. Carefully apply your DPN1 digest sample, which is now 70 microliters, to the center of the gel bed surface. Do not allow the sample to flow around the inner wall of the column. So again, you're trying to hit the middle, right? You don't want to go pipetting on the side and then have it channel down the side of the column. Okay, so do not allow the sample to flow down the inner wall. In step 13, replace the column assembly into the 15 ml tube and centrifuge at 700 times G for 5 minutes. Then remove the column assembly from the 15 ml tube with tweezers. Remove the spin column and collection tube from the rotor, separate them from each other. Your purified sample is now at the bottom of the collection tube. So remember, you added your sample in step 12, and then you spun, and this is where your DNA that you want is just going to go right through the column, because the big molecules, again, are going to go around the beads and come right out. Okay. In step 15, transfer the purified sample from the bottom of the collection tube to a new 1.7 ml orange tube and label it pure PCAS adenine tube. Label this tube with your group numbers and place on ice. Step 16, the volatile staff will collect these samples for speed vacuum evaporation. We will concentrate each sample to 20 microliters, and this will be done by the Courageous staff. Speed vacuum concentration uses a centrifuge that spins your samples with the caps open, pulls the vacuum at the same time. The centrifuge has to be running before you start pulling the vacuum, otherwise it'll suck your sample out of the tube, right? So you get the centrifuge going, your caps are open on the tubes, the instrument pulls the vacuum and also heats up the chamber to about 45 degrees, I think. and that helps the samples to evaporate. We're just concentrating your samples. It's another trick for getting your samples concentrated. So we're going from about 70 microliters down to about 20 microliters. And this is just to, again, concentrate your samples. Okay. So, that's it. Those are the three things that you're doing in three separate experiments. We don't do this very often where we're doing three in one period, but staff will walk you through it. They'll tell you the actual order you're going to do. If we finish by 11:30, which I kind of doubt, come back here. If it's starting to push 12, then we just won't do it. I'll probably come down and make an announcement. Okay? All right, ready, go.